Welcome to this lecture on induced pluripotent stem cells, a topic that has completely revolutionized modern biology and medicine. Today, we are going to explore how scientists have discovered a way to turn back the biological clock on adult cells. We will learn what these cells are, how they are created, and why they hold the key to the future of personalized regenerative medicine. Let us dive in. To begin our journey, imagine you have a piece of clay that you have already baked into the shape of a coffee mug. In the physical world, once that clay is baked, it cannot easily become a plate or a bowl again. For a long time, scientists believed human cells worked the exact same way. They thought that once a cell became a specialized skin cell, it was permanently locked into being a skin cell forever. However, what if you had a magical reset button that could unbake the clay, turning it back into a soft, moldable lump that could become absolutely anything? In biology, this magical reset button actually exists, allowing us to take adult, fully formed cells and turn them back into blank slate baby cells. Now that we understand the concept of cellular time travel, let us look at what these cells actually are. Induced pluripotent stem cells, commonly abbreviated as iPSCs, are a unique type of stem cell. They are generated directly from adult somatic cells, such as ordinary skin or blood cells taken from a patient. The word pluripotent is key here. It means these cells have the extraordinary ability to develop into almost any cell type in the entire human body. They share this incredible adaptability with embryonic stem cells, but as we will see, they are created in a completely different and groundbreaking way. So, how exactly do we press this biological reset button? To create iPSCs, scientists introduce a specific cocktail of 4 genes into the adult cell. These are famously known as the Yamanaka factors, named after the scientist who discovered them. The factors are OCT4, SOX2, KLF4, and c-MYC. You can think of these transcription factors as molecular switches. When introduced, they actively turn off the genes associated with the adult cell's specific identity, like being a skin cell. Simultaneously, they turn on the genes associated with embryonic development. By forcing the expression of these specific genes, the adult cell is completely reprogrammed, losing its previous identity and reverting to a highly adaptable, embryonic-like state. Having discussed the Yamanaka factors, let us visualize this entire process. On the left side of this diagram, you see a standard somatic cell, specifically a skin fibroblast. Notice the 4 arrows pointing into it, representing the introduction of the Yamanaka factors: OCT4, SOX2, KLF4, and c-MYC. The large arrow, labeled cellular reprogramming, shows the transformation of this specialized cell into the cluster of round, unspecialized cells in the center. These are our induced pluripotent stem cells. Finally, look at the right side of the diagram. Because these iPSCs are pluripotent, they can be coaxed to differentiate into entirely different specialized cells, such as a muscle cell, a neuron, or an epithelial cell. This visualizes the complete journey from one adult cell type to another. Now that we have seen how iPSCs are made, we must ask why this discovery is so revolutionary. The creation of induced pluripotent stem cells is so profoundly important that it won the Nobel Prize in 2012, just 6 years after it was first published. Before iPSCs existed, the only way scientists could obtain pluripotent stem cells was by harvesting them from human embryos. As you might imagine, this raised massive ethical concerns and severely limited scientific research due to strict regulations. By allowing us to create pluripotent cells from ordinary adult skin samples, iPSC technology completely bypasses these ethical dilemmas, opening the floodgates for unrestricted, life-saving scientific research. Beyond solving ethical issues, this technology has incredible practical applications for understanding illnesses. Because we can take a small skin sample from a patient and turn those cells into iPSCs, we can then coax them into becoming heart, brain, or liver cells right in a laboratory dish. This means we can study a patient's specific disease safely, outside their body, observing how the illness develops at a cellular level. Furthermore, we can test how their unique cells will react to certain experimental drugs. This allows doctors to find the most effective treatments without ever risking the patient's actual health, revolutionizing how we approach pharmacology. Following up on drug testing, iPSCs offer an even more futuristic application: regenerative medicine. Even more amazingly than modeling diseases, we can potentially use these stem cells to grow entire replacement tissues and organs for patients who need transplants. The most critical advantage here is genetic compatibility. Because these new tissues are grown directly from the patient's own genetic material, their immune system will recognize the cells as their own and will never reject them. If we did not have this technology, personalized regenerative medicine would still be stuck in the realm of science fiction. Instead, it is rapidly becoming an advancing medical reality. While the concept of iPSCs is amazing, the actual process of creating them in a lab is mathematically quantifiable and quite challenging. We measure how well our reprogramming process works using a simple efficiency formula. The formula is E equals N iPSC divided by N initial, all multiplied by 100. Here, E represents the reprogramming efficiency, measured as a percentage. The symbol NiPSC stands for the final number of successfully formed stem cell colonies that we can count in our Petri dish. The symbol N initial represents the starting number of adult somatic cells we attempted to reprogram. If the final number of colonies increases while the starting number stays the same, our efficiency goes up. Conversely, if we start with a massive number of cells but only get a few colonies, the efficiency drops to a tiny fraction. Now that we have our formula, let us apply it to a real laboratory scenario. A research scientist is trying to generate iPSCs to study a rare genetic heart defect. She starts by plating exactly 250,000 skin cells in a culture dish and introduces the Yamanaka factors. After waiting for 4 weeks, she observes the dish and counts exactly 50 distinct, healthy iPSC colonies. We need to find the reprogramming efficiency. We substitute 50 for our successful colonies in the numerator and 250,000 for our starting cells in the denominator. Dividing 50 by 250,000 gives us a raw decimal of 0.0002. Multiplying by 100 converts this to a percentage. The final efficiency is 0.02%. This physically means that out of every 10,000 skin cells, only 2 successfully transformed, highlighting how stressful and inefficient this process is. Because the initial reprogramming efficiency we just calculated is so terribly low, scientists must grow a massive number of the successful cells to use them for therapies. To model this expansion, we use the standard exponential growth formula. The formula is n of t equals n0 times 2 raised to the power of n. In this equation, n of t represents the final population of cells after a certain amount of time has passed. The variable N0 is the initial number of stem cells we start with in our isolated colony. Finally, the exponent n represents the total number of doubling events that occur during the growth period. As the number of doubling events increases, the population grows exponentially, allowing a tiny starting cluster to become a massive supply of biological material. Let us see this exponential growth in action. Our scientist isolates a single iPSC colony containing exactly 400 cells. These cells have a doubling time of 24 hours. She lets them grow continuously for exactly 5 days. First, we must figure out how many times the cells will double. We multiply the 5 days by 24 hours to get the total hours and divide by the 24-hour doubling time. This gives us exactly 5 doubling events, so n equals 5. Now we plug our values into the equation. We have 400 times 2 raised to the power of 5. Evaluating the exponent first, 2 to the 5th power is 32. Multiplying our initial 400 cells by 32 gives us 12,800. This shows that even with low initial efficiency, stem cells can rapidly multiply to provide a limitless supply for medical research. We have covered a lot of ground today, so let us review the key takeaways. First, induced pluripotent stem cells represent a magical breakthrough that allows us to turn back the biological clock on adult cells. By introducing the Yamanaka transcription factors, we can erase a cell's specialized identity and return it to a flexible, pluripotent state. Second, while we saw mathematically that the reprogramming process is highly inefficient, the few cells that do succeed can multiply exponentially to create an endless supply. Finally, and most importantly, this technology completely bypasses the ethical dilemmas of embryonic stem cells. It opens the door to a future where personalized, patient-specific replacement organs and targeted disease models are a standard part of medical care. You now understand the fundamental science behind one of modern biology's greatest achievements.